Hi everybody, Brian here, and welcome to this next section on genetics and natural selection. Today we'll be talking about how genetics and natural selection form the foundation for the process of evolution to occur. So for evolution to occur, there are a couple of conditions that need to be met. First, there must be genetic variation among individuals in a population, so there's some kind of difference in the genetic makeup for evolution to act on. Organisms must be able to reproduce and have a systemic way for passing that genetic information to future generations. And finally, natural selection that favors certain traits or certain genetic makeups that give an individual a competitive advantage over other individuals in that population, or an introduction of new genetic information into a population, such as migration. So we'll start by tackling this first point, and before we get into all the nuances of genetic variation, we'll start with the very basics of essentially what your genetics are. So most of you have probably heard about DNA at some point, and that it is unique to each individual. So unless you're a twin, no other person will have the same genetic makeup as you. So DNA stands for deoxyribonucleic acid, and we can see here it is this double helix structure, kind of like a twisted ladder. And it's made up of these four basic building blocks called nucleotides, or base pairs. These nucleotides are adenine, guanine, thymine, and cytosine, which are represented by the first letter of their name. And you can see that in the picture there are A's, T's, C's, and G's. One important thing to note is that adenine and thymine will always bind together, and cytosine and guanine will always bind together, so you'll never see an AG or an AC binding. And so this sequence of base pairs forms a unique sequence that we can see represented in this picture at the bottom. And this entire sequence is known as the genome, and it contains all of the biological instructions that an organism needs to build itself, to maintain, and to reproduce. So it really is the blueprint for life. And just to give you a reference on how much information there is in the genome, for the human genome, there are about 3.2 billion base pairs. So if you were to open up a Word document on your computer and type out the entire sequence of your genome in regular font, it would be enough data to fill up an entire CD disk. And so that's an incredible amount of information that's stored in each one of your cells. And now that we're familiar with what DNA is, its structure, we can zoom out a little bit and look at the next functional unit of DNA and our genetic makeup. So genes are the basic physical unit of heredity. So when we talk about things being passed on through the generations, we're talking about usually changes in genes. And these are entire portions of the DNA whose sequence codes for a protein or some functional product in the body. In the human genome, there are an estimated 20 to 25,000 genes. And in this picture, we're visualizing the steps going from your DNA strand to a working product like a protein. So your body, you have this DNA sequence right here, and your cells will then make a copy of that, a complementary strand in the form of RNA, which we don't have to get too much into. And from that, there are these things called codons, which are groupings of 3 nucleotides that will encode for an amino acid that will get added to a chain which will eventually form a protein. And depending on the letters, the sequence of letters in that codon, that will tell you what amino acid is getting added. And so in this case, you can see here from the M on this amino acid, that methionine, abbreviated by MET, is the first amino acid. And then you'll move along those different codons and tack on amino acids, forming this chain, which will eventually form a fully functional protein that can now perform cellular work within the body. And those genes are arranged on chromosomes. So kind of zooming out a little bit, we have your individual nucleotides here, and then entire sections of that are genes, which are then located on the chromosomes, of which you get one from each parent. So you'll have 2 copies of each gene, and then those are stored in the cells. However, it's important to note this example is for a eukaryotic cell. It's a little different based on whether or not you're a prokaryote or a eukaryote, which we'll get into that in just a little bit. But for now, we'll have you complete the Evidence for Evolution Molecular Evidence Lab simulation before moving on. Now we'll move on to our second point here, where now that we've established that how genetic variation exists in the genome, you have to be able to pass that genetic information to future generations. And the mechanism for this is cell division, where a cell will copy its DNA and create 2 identical daughter cells. And this process is a little different depending on whether or not you're a prokaryote or a eukaryote. And in case you maybe learned about this way back when, or it's new to you, prokaryotes are much simpler cells. They have no nucleus or any type of cellular organelles. They have circular DNA, which means it's just one big connected circle of DNA, and they're always unicellular. So an example would be bacteria or archaea. And with eukaryotes, these are more complex cells. The DNA is stored in a nucleus, and we have all these other organelles within the cell. And the DNA it's linear, so it has a start and a stop point, and it can be unicellular or multicellular. And this includes plants, animals, fungi, protists. Pretty much any living organism that you can see with your naked eye is a eukaryote. So we'll break down the different ways that prokaryotes and eukaryotes go through cell division. Prokaryotic cells divide through a process called binary fission. We have this picture on the right that is visualizing this process, and I've just kind of summarized those steps. So when the cell is ready to duplicate itself, the DNA is replicated or copied. And as the DNA is being copied, the cell is going to elongate to ensure that you have enough cell material for like 2 fully functional cells. That replicated DNA will be separated into opposite sides of that elongated cell, and you end up with 2 genetically identical daughter cells. So you've essentially copy and pasted this original cell. And if you think back to our requirements for evolution to occur, you have to have genetic variation. And by this process, you're never going to just produce differences because you're just copying. But there are ways for the bacterial genome to change naturally. One of those ways is through random mutation. And this can also occur in eukaryotes, so just keep that in mind moving forward. And random mutation occurs when your cell is copying the DNA and it makes a mistake. So most of the time, cells do this with really, really high accuracy, but they do make a mistake. So in this example, we have our original DNA sequence right here. We have CAT being repeated over and over again. And each of these codons, the CAT codon, encodes for this his amino acid. Now in this example below, the original DNA sequence has a mutation at this point here, where instead of an adenine, we have a cytosine. And that actually results in the incorrect amino acid being inserted here. You changed the codon enough to where it now encodes for a different amino acid. And the effect that this can have is quite variable. It can be negative, where either the protein is no longer functional or it doesn't function as well as it should. It can be neutral, where you change that sequence but it still has the same amino acid. Or it can actually be advantageous in some scenarios. So maybe that protein operates just a little more efficiently and provides some sort of advantage. So random mutation is one way. Transduction is another way, which is the transfer of genetic material from some type of bacteriophage, like a virus, to a new bacteria. So in this example, a virus will infect a bacteria with its own DNA, hijack that bacterial cell to produce more of the virus, and in that process, it might pick up some of the bacterial DNA. It will then go out and find a new bacteria to infect, but instead of injecting the viral DNA, it will inject the DNA from the previous bacterial cell, which can then be incorporated into this new bacterial cell. So that's another way that you can get genetic information, new genetic information into a cell. A third method is conjugation, and this is the transfer of genetic material from bacteria to bacteria through the formation of a direct connection between these two cells. So certain bacteria are able to form this physical connection and directly transfer their genetic material to another bacterial cell. And finally, and this one's actually very important for things like antibiotic resistance, is natural transformation, where a bacterial cell picks up genetic information from the environment. And this is important because that environmental DNA doesn't even have to be alive. So let's say you have sanitized your countertop and you've killed all the bacteria there, you've destroyed its cellular structure, but it still has some of its genetic material is present in a non-living form. Now, a new bacteria that is still alive could pick up that genetic material from the dead bacteria and incorporate it into its own genome. And you'll see that in your lab simulation on antibiotic resistance. But before that, we'll now talk about how eukaryotic cells go through cell division. And they do this through a process called mitosis or meiosis. Here we have a representation of your normal cell cycle for a eukaryote. Most of this is spent in what is called interphase, during which time the cell is growing. It's just kind of doing normal cell things. When it becomes time to divide, it will copy all of the chromosomes. It'll make a copy of all of that genetic information within the cell and prepare for division. Now, mitosis occurs in all cells except for your reproductive cells. So the cells in your skin, your mouth, your muscles, all of those are going to go through mitosis, whereas cells involved in reproduction, like sperm and eggs, go through meiosis. And the two processes are similar in concept, but there are some key differences between the two that we'll talk about. So let's start with mitosis. And this produces two genetically identical diploid daughter cells. Diploid meaning that you have two copies of the chromosomes, one from each parent. So in this picture, we'll start on the left in interphase. We can see that all that genetic material is kind of unwound in this big stringy mess. And that's because for the cell to be able to access that genetic information to make the protein products, it has to be accessible, so it has to be unwound. So once we finish interphase, we're ready to divide. We copy the contents of the cell, and move into prophase, where the nuclear membrane that was enclosing that DNA has now dissolved, so that blue line representing the nucleus has now gone away. And the DNA has now condensed. So in order to move this genetic material around, it's much easier if it's all condensed down into these easily movable units. And then we also have these structures called centrioles that move to the opposite ends of the cell. Then we move forward into metaphase, where those condensed chromosomes will line up end to end. So they're two ends touching each other like this in the middle of the cell. And these spindle fibers extend out from the centrioles and grab the chromosomes at what are called centromeres. In anaphase, those spindle fibers will contract, and that pulls apart these duplicated chromosomes to either end of the cell. And then finally in telophase, the nucleus will reform around the genetic material on either side, and the cell will pinch in the middle, forming two identical daughter cells. And a common acronym for this process to help you remember it is IPMAT. So if you're trying to remember the order of these stages, just remember IPMAT. Or if you're a mnemonic device person, which certainly helps me, we have these two mnemonic devices: I passed my algebra test, IPMAT, or idiot, pass me another tequila, whichever one helps you remember the order of these stages. And so now we'll talk about mitosis, and we'll see that it's fairly similar in concept, but there are some really key differences. Now, meiosis will produce 4 genetically different haploid daughter cells. Haploid meaning they have half the amount of genetic material. But we can see that the stages are basically the same. So there's prophase, metaphase, anaphase, telophase. But one of the major differences is that there are actually 2 rounds of cell division as opposed to just the one. So they're divided up into meiosis 1 and meiosis 2. Also, they left out interphase in this picture, but interphase is still occurring in meiosis. And we mentioned that this produces 4 daughter cells that contain half the number of chromosomes. So let's just work through this, this process. In prophase 1, similar thing happens where the nuclear envelope has gone away, and you have the centrioles moving to either side of the cell. In metaphase, this is where one of the key differences lies, in that in metaphase, in mitosis, the cells are lining, or the chromosomes are lining up end to end, but in metaphase 1, the chromosomes are lining up next to each other. So they're lining up adjacent to each other, not end to end. And then the spindle fibers will attach to the chromosomes in the same fashion. Anaphase, very similar. It'll pull those entire chromosome pairs to either side of that cell. Telophase will then form two different copies of that cell. But then a second round of cell division occurs on these 2 cells that were just produced. So they're going to go right into another round of cell division. We see the same thing in prophase. That nucleus is going to disappear. And now metaphase here looks similar to how it did in mitosis, where our chromosomes are now lining up end to end. The centrioles the spindle fibers and the centrioles will grab those chromosomes at the centromeres, pull them to either side of the cell, and then those cells will cleave in the middle, and the product that you get is 4 daughter cells that contain half the number of chromosomes. And this is because you have half the genetic material provided by one parent and half the genetic material provided by another parent, and they combine to provide the full genetic makeup that is required for life. Now, I mentioned that random mutations occur in eukaryotes. However, the other three methods of getting genetic change within the cell don't occur in eukaryotes. So there's no transduction, conjunction, or natural transformation. However, there is something called crossing over that occurs in prophase 1, which is really important for increasing genetic variation. So crossing over is the exchange of genetic material on homologous chromosomes, or chromosomes that are the same. And this occurs in prophase 1, as I mentioned. And what happens is, during this time, the chromosomes are lined up side to side. And the parts that are next to each other can sometimes swap bits of that chromosome. And what that does is it changes the arrangement of alleles on the chromosome. Alleles are different versions of the same gene. And this functions to increase genetic variation in the offspring, even if you don't have changes to the genes through something like random mutation. And so this is really important for increasing some of that genetic variation in offspring. And at this point, we'll have you complete the human genetics chromosomal inheritance during meiosis and genetic inheritance lab simulations before moving on. And finally, we'll talk about our third point, natural selection favoring genetic traits that confer a competitive advantage. So natural selection is a mechanism for evolution. So it's a force that essentially drives individuals to change. And this is caused mostly by resource limitation or predation that creates competition among individuals. So in any natural system, you're going to be limited by the amount of food that you have, the amount of space that's there, nutrients, basically anything that an organism might need to survive will be limited. And so organisms that are better adapted to take advantage of their environment are more likely to survive and reproduce and pass their genes on. So if there's an organism that has a slightly better sense of smell or is maybe a little bit faster, they will survive, and then their genes will be passed along to the next generation. And then genes passed down over generation cause the population to change and diverge over time. So in this picture, we have this classic example of the Galapagos finches, which Charles Darwin observed and kind of used for this concept, where we have this really great diversity of finches that likely arose from a single type of bird. And so if we look at this ancestral bird that ate seeds off the ground, maybe one season seeds were really hard to find. And so birds that were able to take advantage of a different food source, such as insects, were able to go after a new food source and kind of find their own little area of this ecological niche and survive and reproduce. And even within the seed eaters, maybe some of the seeds were really large and hard to break open. And if some individuals had a slightly larger or more powerful beak, they were better at getting to those harder to access seeds. And so this drives these species to change and take advantage of the environment in different ways that allow them to survive and reproduce. So how do we know if a population is evolving? And we can examine this with a concept called the Hardy-Weinberg equilibrium. If a population is not evolving, then that population is in Hardy-Weinberg equilibrium, meaning that the frequency of alleles, or the different versions of genes in a population, is not changing over time. And that is represented by this equation here. P squared plus 2pq plus q squared equals 1. And these are the genotype frequencies. So in your previous section, you talked about genotypes and alleles and dominance and recessive of certain alleles, and that's going to come into play in this section here. So in this equation, these are your genotype frequencies, And then in this section, these are the frequency of the actual alleles themselves. And they have to equal up to 1 because the sum of all the alleles equals the total. So that equals 1. In this equation, homozygous dominant is represented by one of these squared terms. Heterozygous, so having a mix of those two alleles, is represented by this 2pq. And then homozygous recessive is represented by the other squared term. And there are a couple of assumptions that we make for a population to be in Hardy-Weinberg equilibrium. So we assume that there's no mutation, so that the genetic makeup is not changing. Random mating. This is a bit of a more complicated assumption, so we'll just kind of gloss over that. No gene flow. Again, this is just assuming that there's no changes happening to the genes in the population. Infinite population size. There are issues that you run into when you have really small populations that are reproducing. And no natural selection. So in a population that's not evolving, we assume that all of these things are happening. Obviously, in any real natural system, you know, at least some of these things are likely to be happening, but this is a very hypothetical situation that allows us to investigate whether or not a population is evolving. And this probably makes 0 sense to you right now, so we're going to go ahead and run through an example of how you would examine this with the Hardy-Weinberg equilibrium. So in this example, we have a population of mice with 2 different color variants. We have black fur, that is the dominant gene, represented by capital B, and then we have white fur, which is the recessive gene, represented by the lowercase b. 70% of this population has black fur. However, we don't know their genotypes. Because black fur is dominant, you can either be homozygous dominant or you can be heterozygous. And so we don't know their genotype, and so we don't know the allele frequency. 30% of these mice have white fur, and because the white fur is the recessive gene, we know that the genotype has to be little b, little b. But we still don't know the allele frequencies, but using this Hardy-Weinberg equation, we can calculate them. So we have the original equation here, p squared plus 2pq plus q squared equals 1. We'll start by calculating q, which is the allele frequency of this recessive gene. And to do that, we take this q squared term from the Hardy-Weinberg equation. And that represents our our little b genotype. And we know that it's 0.3 because it's 30%. And to calculate what the allele frequency is, we simply take the square root, and we find that the allele frequency of the little b is 0.55 or 55%. From there, it's a fairly simple calculation to calculate the frequency of our dominant gene, because we simply subtract it from the total, the entirety of the allele frequencies, and find that the dominant allele has a frequency of 0.45 or 45%. And from here, it's simple plug and chug. So to calculate the genotype frequencies, we put 0.045 for the p squared. To get the heterozygous, we multiply 2 times 0.45 times 0.55, the allele frequencies of both the dominant and recessive, and then we do the square of the allele frequency for the recessive gene, which is 0.55. And when you calculate all that out, we get 0.2, 0.5, and 0.3, which adds up to 1. So our math checks out. And what we get is a genotype frequency for the dominant, homozygous dominant of 20%. The heterozygous genotype is 50% of the population. And then 30% of the population is this recessive white genotype. But to know if this population is evolving, we have to examine it over several generations. So let's look at this next generation where we have now 90% have black fur and only 10% have white fur. So same thing, we can't know what the genotypes for the black fur individuals are, but we do know the genotype for the white fur. And in this case, they make up 10% of this population. And so we'll just run through the same calculation where we figure out what the allele frequency is for the recessive gene, which is 0.32 or 32%. We then calculate the allele frequency for our dominant gene, which is 0.68 or 68%. Again, we plug and chug, and we can find the new genotype frequencies of the second generation. And if we compare them, we see that the genotype and allele frequencies are changing. And that would tell us that this population is evolving. And then we could go in further and figure out which of the assumptions are being violated. You know, are you having mutations, gene flow, stuff like that. And to just give you more of a kind of visual representation of what this might look like in a natural population, let's imagine that we have this population of snails that lives on this sandy beach, and we have a starting population of snails with a variation in shell color. So you can be brown, which blends in more with the sand, or you can have a colorful shell. Now this snail population will eventually grow, and they are preyed upon by these birds. And the population will continue to grow until a certain point where resources are now limited. And so maybe these snails have to travel further to find food, or there's less space for them to take shelter and stay hidden from these birds. And the birds are more easily able to spot those individuals with colored shells. So in this picture, the ones with the bright shells have been made larger just to represent that the eagle or the birds are able to see them better. And so they're preyed upon more often and removed from the population. We see here that the ones with the colorful shells are being removed more than the ones with the brown shells. And what you end up with is a population that has shifted from kind of more of an equal distribution of light, or a kind of this brown color and coloration, to most of the population having just brown shells. Now you're always going to have some genetic variation within the population, and so we see that there still are individuals with colorful shells, but the proportion of them is much lower because the ones with the shell that blends in with the sand are able to survive, reproduce, and pass their genes on to the next generation. And with that, we'll have you complete the final 2 lab simulations: natural selection antibiotic resistant bacteria and natural selection insects, and you're done. Hopefully this will help you to understand the simulations going forward. This is a very difficult topic to grasp. So if you have any questions, things you want clarification on, please reach out to your TA, and we would be happy to elaborate on some of these concepts. All right, and thank you for listening, and good luck.